Welcome to DigiDig, where we uncover the past and explore the future.
Look out at the modern North Sea, and you will mostly see a bleak, freezing expanse of gray water. It feels impenetrable. But if you drain that water and wind the clock back to 10,000 BCE, you won't find an ocean. You'll be standing on Doggerland. This was a massive continental bridge stretching from Britain to mainland Europe, covered in birch forests and woven together by massive river networks. It was a highly productive estuarine environment, and it belonged to the Mesolithic hunter-gatherers. These humans were apex predators, thriving in the wetlands alongside massive fauna like wild boar, aurochs, and giant elk. As the last glacial maximum ended, rising global temperatures began melting the Great Laurentide and Fennoscandian ice sheets. Millions of gallons of fresh water poured into the oceans, and the rising sea slowly began to swallow Doggerland's low-lying plains. That slow drowning was interrupted by a sudden, violent fracture. Around 6200 BCE, a massive submarine landslide known as the Storegga Slide ripped away a huge chunk of the Norwegian continental margin. 3,500 cubic kilometers of debris collapsed, triggering a mega tsunami. Waves several meters high slammed into the remaining landmass, sweeping deep into the interior and devastating the surviving coastal communities. Within a few centuries, Doggerland was entirely submerged. An entire epoch of human history, along with the very ground they walked on, was swallowed whole, locked away as a dark ghost world beneath the ocean. Today, that prehistoric landscape sits beneath dozens of meters of dark, freezing muck, entirely isolated from human reach. The only modern contact with this drowned world comes by accident. Commercial Dutch fishing trawlers routinely drag heavy nets across the submerged plain. As these ships dredge the ocean floor for fish, their rigging occasionally scrapes into the sediment of the early Holocene, pulling isolated fragments of Doggerland up to the surface. Among the most frequent items pulled from the nets are these slender artifacts. They are barbed points, shaped from raw bone and antler. These jagged profiles were designed to wedge deeply into the flesh of their prey. The Mesolithic hunters used them as lysters for striking fish in shallow lagoons, or as projectile tips for hunting large land herbivores. Across the entire submerged continent, these points appear as the primary means of securing protein, the specialized hardware that kept human populations alive for 5 millennia. Curious tone. Archaeologists understood how the tools worked, but they faced a massive blind spot. They had no way of knowing what specific animal these bones belonged to. These serrated fragments are some of the only physical evidence left of life in Doggerland, yet their biological origin remains invisible to the naked eye. Normally, identifying a bone tool relies on simple biology. Zoo archaeologists look for natural structural markers, the specific shape, ridges, or surface textures unique to a specific animal. However, the Mesolithic hunters systematically removed those identifying features during the manufacturing process, a sequence of steps known as a chaîne opératoire. They scraped, ground, and heavily carved the bone. This modification completely stripped away the periosteal surface, the outer layer of the skeleton that holds all the diagnostic biological markers. What human hands didn't destroy, the ocean finished off. These artifacts spent millennia at the seabed, subjected to extreme tectonic pressures. A relentless combination of abrasive sand currents and marine mineralization created a smooth, hardened finish known as a marine polish. Human ingenuity and harsh ocean currents worked together perfectly. By the time these weapons hit the trawler nets, the outer identity of the raw material was entirely physically erased. When the external surface fails to provide answers, scientists must look deeper. For decades, researchers relied on early microscopes to study the bone's internal tissue. This animation shows exactly what they saw. Because the weapons required high strength, hunters carved them from thick cortical bone, which relies on a dense cellular structure known as Haversian tissue. As you can see, the size, shape, and clustering of those microscopic cellular structures overlap almost entirely among large mammals. Through a lens, the cellular makeup of an elk bone looks virtually identical to a red deer bone. This created a massive, incredibly frustrating bottleneck for the scientific community. A century's worth of recovered artifacts sat in museum drawers, taxonomically useless. The true identity of these weapons was scrubbed away by time, human tools, and the weight of the sea. They were perfectly silent artifacts, sitting in the dark, forcing researchers to simply guess what.
10,000 years ago, a massive, unbroken land bridge connected Great Britain to the European mainland. This was Doggerland, a thriving Mesolithic landscape of pine forests and river basins that was entirely swallowed by rising sea levels at the end of the Ice Age. For decades, Dutch fishing trawlers have been dredging up the remnants of this drowned world. Among the most common finds are these barbed osseous points. They are slender, carefully carved tools covered in sharp serrations, designed to lodge securely into the flesh of prey. Archaeologists long assumed these weapons were carved from the skeletal remains of large, common herbivores available on the prehistoric plains, specifically red deer and elk. The problem is, verifying that assumption visually is impossible. The manufacturing process involves intense scraping and grinding that strips away the bone's outer diagnostic layers. Add 8,000 years of marine taphonomy, abrasion from sand and salt water, and the original morphological markers are completely erased. This physical degradation created a massive blind spot in our understanding of Mesolithic technology. Because the visual history of these weapons was destroyed, researchers were left guessing at the true origin of the materials. When macroscopic identification fails, the standard archaeological procedure is to look inside the bone using microscopic histology. However, at the microscopic level, humans and large mammals share a highly similar biological architecture. Adult human cortical bone and the bones of large cervids are built using the exact same structural foundations. This chart compares the microscopic structural features of human bone against those of large mammals like deer. Looking closely at the primary tissue types, both species exhibit dense, closely packed haversion systems, making them indistinguishable on a broad cellular level. Even taking precise measurements fails to provide a clear answer. The osteon diameters for both humans and cervids overlap entirely at approximately 50 micrometers. The one feature that could definitively tell them apart is the internal medullary surface. But hunters intentionally carved that delicate, spongy web away to ensure the weapon shaft was built strictly from the thickest, strongest part of cortical bone. Visual morphology and microscopic histology are entirely incapable of solving this taxonomic mystery. To find the truth, researchers had to abandon visual evidence completely and rely on molecular forensics. Collagen type 1 provides the molecular data necessary for species identification. This structural protein is incredibly durable, persisting in the archaeological record long after cellular DNA degrades. Its genetic coding varies significantly between different animal taxa. Accessing that collagen requires a delicate extraction process that avoids destroying the artifact. Researchers scrape just 10 to 30 milligrams of material from a non-diagnostic edge of the weapon. The bone dust is placed in a mild chemical solution called an ambic buffer. This process gently dissolves the rigid mineral matrix of the bone, successfully releasing the trapped collagen inside. The liquid sample is then heated. This denaturation phase causes the tightly coiled collagen triple helix to unwind, separating into 3 distinct, straight polypeptide chains. Next, scientists introduce trypsin, a highly specific digestive enzyme. Trypsin acts like a pair of chemical scissors. It is programmed to cleave the polypeptide chains strictly after encountering 2 specific amino acids, lysine and arginine. Through extraction and digestion, scientists transform an unidentifiable piece of solid bone into a liquid soup of genetically coded peptide fragments. The material is now ready to be measured. The researchers now have the isolated genetic fragments, but these pieces are far too small to be seen under any microscope. Instead of looking at them, they have to weigh them. This requires a MALDI-TOF mass spectrometer. The liquid sample is deposited onto a target plate, which acts as a launch pad inside a long vacuum tube. During the ionization phase, a powerful laser strikes the chemical matrix. This vaporizes the sample, instantly converting the liquid into a cloud of charged ionized gas particles. An electric field is then activated, pulling the charged peptide fragments out of the cloud and accelerating them straight down the vacuum tube.
This is the time-of-flight mechanism. Physics dictates the sorting process. The lighter peptide fragments fly rapidly down the length of the tube, while the heavier, bulkier fragments lag behind. A detector at the end of the tube records the arrival time of each fragment. That flight time translates directly into a precise mass-to-charge ratio. Because the original amino acid sequence varies by species, the trypsin scissors cut the chain into different lengths for different animals. The resulting cluster of masses forms a distinct, unmistakable mathematical barcode. This is the power of the Zooms protocol. It bypasses 8,000 years of physical abrasion and marine decay entirely, relying exclusively on the indestructible physics of molecular weight. In 2020, researchers at Leiden University applied this molecular technique to a collection of North Sea artifacts. One specific barbed point, catalogued as specimen P03, yielded highly unexpected results. When the spectrometer plotted the data for sample P03, researchers knew exactly what a normal baseline should look like. They expected to see the standard biomarker peaks associated with red deer and elk, confirming the long-held assumptions of the archaeological community. But the initial hypothesis failed. The peptide masses from sample P03 did not align with any known prey species from the Mesolithic plains. The empirical data revealed highly specific anomaly markers, registering sharp peaks precisely at the 1400, 1500, and 2800 marks on the x-axis. When run against the reference database, that exact combination of peaks provided a single, absolute match. This molecular fingerprint belongs exclusively to Homo sapiens. These artifacts were not loose, accidental fragments of human remains. They had been intentionally shaped, sharply barbed, and intensely curated for violent application as hunting weapons. This definitive molecular proof transforms these artifacts from simple hunting tools into evidence of an intense cultural practice, where the equipment of the hunt was inseparable from the remains of the dead. The discovery immediately poses a difficult question. Who were the people whose remains were systematically weaponized? This chart visualizes the radiocarbon and stable isotope data from the extracted collagen. Radiocarbon analysis dates specimen P03 to 8,200 uncalibrated years before present, confirming this persistent regional practice spanned nearly a millennium in Doggerland. To understand their diet, researchers analyzed the nitrogen-15 column. High concentrations of nitrogen-15 indicate a heavy reliance on animal protein. These individuals sat at the absolute top of the food chain, living as apex terrestrial hunters before their bones were repurposed. And they were not repurposed out of desperation. Red deer antler is mechanically superior to human cortical bone for absorbing impact. Furthermore, processing a human cadaver to isolate the bone requires significantly more effort than using the leftovers of a fresh kill. Because human bone is inferior in strength and harder to harvest, the choice to use it was cultural rather than economic. The decision was driven by identity, not utility. For the hunter-gatherers of Doggerland, the boundaries defining a person, an animal, and an inanimate object were entirely fluid. Anthropologists characterize this worldview through the lens of animism, where objects are perceived as sentient tools, possessing a living essence and distinct personhood. By carving a spear point from the femur of a deceased elder or a revered hunter, the surviving kin could imbue the weapon with the skill of their ancestor, keeping the dead active in the daily survival of the tribe. These points functioned as ancestral relics, allowing the deceased to provide for the living long after their death. This intense relationship with the dead likely deepened as their world collapsed. As Doggerland was slowly swallowed by the sea, tribes losing their ancestral burial grounds may have been forced to transform their dead into portable relics they could carry with them. The hunting technology of Doggerland was physically woven from the remains of the dead. It is a complex reflection of human psychology and survival, one that remained completely invisible.
Thank you for watching.